Hi, good evening everyone. Today we'll take the second problem solving session for the course Biochemistry. My name is Shrija Ghosh. I'm a PMRF at IIT Kanpur and I'll be taking the problem solving session for this course. The course instructor is Professor Swagata Dasgupta. You might have seen the lecture videos of her already. So I'll be here to clear any doubt regarding your week 2 assignment. So I'll be solving certain questions related to your week 2 assignments, and at any point if you guys have any doubt you can stop me and ask. Okay, you can either write in the chat box or you can unmute and ask your question. Okay. Okay. So we'll start now. So I'll start with the first question. A student synthesizes two novel amino acids A and B whose structures are given. So we have two structures given here. The hypothetical hydrophobicity is calculated according to the Kite-Doolittle scale, which set represents the correct hypothetical values. So we have different values given for the hydrophobicity scale or the Kite-Doolittle scale. And we have to tell which of the values could be correct for the given structures. Okay, so we have two structures here. Can anyone tell me what is the difference between two structures? Like, can you see any significant difference between the two structures? Any of the physical or chemical parameters? There's a very significant difference between the two structures if you see carefully. So in one of the structure, if you see, there are NH groups present in the ring structure and the other structure has benzene rings attached. So what does these nitrogen do? So nitrogens are highly electronegative atoms. Okay. So what can nitrogen do? Nitrogen can happily make hydrogen bonds. Okay. So we have here two structures and we were asked the Kite-Doolittle scale, the hypothetical values for the Kite-Doolittle scale. So now what is the scale? The scale is basically the hydrophobicity scale. Okay. So let's say I have any molecule, and if it is hydrophobic, it will have a positive value, and if it is hydrophilic, it will have a negative value. Okay, so here we have two structures. So what do you think, which one of this is hydrophobic and which one of this is hydrophilic? Can anyone answer? Which structure is hydrophobic and which structure is hydrophilic? I have given you the hint already. Anybody? You can either write in the chat box or you can unmute. No one? Okay. So basically, if you look into the structure, I told you nitrogens can make hydrogen bonds. So these nitrogen can happily make hydrogen bonds with the hydrogen of water molecules. Okay. So basically, this molecule is hydrophilic. Okay. And this molecule is hydrophobic. Why this molecule is hydrophobic? Because the molecule has a benzene ring attached to it. Benzene ring, it's a conjugated system. It cannot make hydrogen bonds with the water molecules. So it is hydrophobic in nature. Okay. Now, what are we saying here? We are saying that hydrophobic amino acids have a positive value and hydrophilic amino acids have negative value. So, if you see, I am saying A is hydrophobic. So, hydrophobic should have a positive value. So, option A or B are there, and then hydrophilic should have a negative value. Only option A has positive for structure A and negative for structure B. So, the correct answer is option A. Okay. This is clear. So, basically this hydrophobicity scale or the Kite-Doolittle scale are the values that define the relative hydrophobicity or the hydrophilicity of a amino acid residue. So, if a residue is hydrophobic, it will have a positive value. If the residue is hydrophilic, it will have a negative value. Okay. So, I have a list of amino acids, all 20 amino acids, and you can see the hydropathy index of all these amino acids here. So, if you see the hydropathy index of all these amino acids, so non-polar amino acids, what do you mean by non-polar amino acids? They are basically hydrophilic or hydrophobic? Non-polar amino acids. Come on guys, answer. Are you able to understand anything? If you are not able to understand anything, stop me and ask. Do you understand what hydrophobic and hydrophilic means? Anybody? Okay. So, hydrophobic means it does not like water, it will not dissolve in water. Okay, and hydrophilic means it likes water, it will dissolve in water. So, if a molecule can make hydrogen bonds with
Hydrophilic
water,
means positive value.
sorry?
Hydrophobic means positive value and hydrophilic means negative value.
Yeah, that is true. But what in general hydrophobicity means? Means it does not like water, right? It will not dissolve in water, right? Any molecule that will dissolve in water will be hydrophilic. Okay. So, here if you see non-polar amino acids means hydrophobic, right? So, all non-polar amino acids have a positive value except 2. So, there is an exception among the non-polar amino acid. If you see the hydropathy index here for glycine and for proline, even though they are non-polar amino acids, they have a slightly negative hydropathy index. Why? For glycine, what is the side chain of glycine? The R group of glycine. What is the R group of glycine? In the previous session, I have told you to study the structure of amino acids. I have told you how all the structures of amino acid, and I have told you to try to remember those structures. Anybody remembers the structure of glycine? The R group of glycine. Glycine is the smallest amino acid.
H.
Yes, the R group is just H. Okay. So, if you see the structure, basically the alpha carbon has the carboxylic group, the amino group, and 2 hydrogens. So, it's a very small amino acid. So, it does not really affects the hydrophobicity or hydrophilicity of a protein. So, if glycine is present in a peptide chain or in a protein, it does not really affect the hydropathy index of the whole peptide. So, if you see the value is not very like the value is very close to 0 only, it is towards the positive scale, but still it is slightly negative. For proline, the reason is, if you remember the structure of proline, the amino group is not free. Okay, since the amino group is not free, it makes a rigid structure. Okay, for proline it's not a very flexible structure because the R group and the amino group are attached together, right? So in that case also it kind of differs from the general rule of hydropathy index. Okay, so for glycine and for proline it's an exception. Other than that, if you see, all non-polar amino acids are hydrophobic in nature. Since they are non-polar, they don't have any polarity, so they are hydrophobic in nature. They do not like water, and thus they have a positive hydropathy index, positive values for on the Kite-Doolittle scale. Okay, similarly, if you see for, oh sorry, I haven't written the values here. If you see, I'll change this part. So if you see for aromatic groups, most of the, so serine, okay, tell me if I have not written the value. Tell me, see, will serine have a positive value or negative value? Anyone can tell me serine, whether serine is a hydrophilic amino acid or a hydrophobic amino acid?
Hydrophilic. OH
Yes,
group is there.
yes, OH group is there. Very good. So if OH group is there, it is hydrophilic because it can make hydrogen bonds with water. And since it is hydrophilic, the hydropathy index would be negative. Okay, because for hydrophilic molecules, we have negative hydropathy index. Okay, so for serine, for threonine, cysteine, most of these amino acids will have a negative hydropathy index. Okay, okay, I've written here the values. Okay, so serine, threonine, cysteine. So cysteine also has slightly positive because again the mostly what happens in a protein when cysteine is there, the cysteine forms disulfide bond with itself. Okay, and thus the SH group, the sulfhydryl group, is generally not free, and thus the hydropathy index is towards the positive, towards the hydrophobic side. Okay, rest all the charged amino acid, if you see, since there is a charge, it can easily dissolve in water. It's a very polar molecule, and then these charges are basically making the molecule hydrophilic and thus has a negative hydropathy index. Okay, so hydrophobicity scale is clear, I hope. So basically any molecule, if it is hydrophobic, it will have a positive value for on the hydrophobicity scale or the Kite-Doolittle scale, and if it is hydrophilic, it will have a negative value. Okay, so next question, which of the following statement is false? So there are 4 statements given. Non-polar amino acid residues remain in the interior of a membrane. Non-polar amino acid residues acts as surface lipid anchor in a membrane. Polar neutral amino acid residues form the hydrogen bond network in a globular protein, and polar charged amino acid residue prefer to remain on the surface of a globular protein. So which of the statement is false? Okay, so we have to find the wrong statement in this. Anyone wants to answer the question?
B. Non-polar amino acid act as surface lipid.
Okay, so Joseph is saying option B. Let's see the correct answer. So non-polar amino acids acts as surface lipid anchor. So what is membrane protein? You said option B, so we'll start with this only. So if you see a membrane protein is basically proteins that are anchored in the membrane. Okay, so here if you see the figure, this figure, okay, these are the membrane protein. Either it can span the membrane or it can be a part of the upper or the lower part of the membrane. Since membrane is a lipid bilayer, so you have polar head group and then hydrophobic chains. Okay, so there is a polar head group and hydrophobic chains are towards inside. So if a protein is there which is, sorry, So if a protein is there, they have said non-polar amino acid residue acts as surface lipid anchor in a membrane. Okay. So what do you mean by non-polar amino acid? I just told you non-polar means hydrophobic. Okay. So for membrane proteins, hydrophobic part would be anchored in the membrane, because the part that we are seeing here, this is basically the hydrophobic part. Okay, the inner part of the membrane is hydrophobic. So the only non-polar amino acids can interact with these hydrophobic chains. So that's why generally this is a globular protein where the polar amino acids are outside, towards the outer side, because the polar amino acids are basically interacting with the polar amino acids are basically interacting with your outer hydrophilic environment. Okay, so let's say the outer environment is water. So if a protein has to interact with water, it needs polar amino acids. It needs hydrophilic amino acids on the outer side of the protein. If a protein is interacting with a hydrophobic environment, like in case of membrane proteins, in such cases the protein needs hydrophobic residues on its surface, so that it can interact with the hydrophobic chains of the membrane. Okay, so Joseph, is this clear? Non-polar amino acid residue will act as the
Yes,
surface lipid anchor in a membrane protein.
it's
Okay,
fine.
so option C is polar neutral amino acids amino acid residues form hydrogen bond network in globular protein. This is also correct, because in a globular protein, I told you, on the surface we have polar amino acids, we have hydrophilic amino acids, and they can form hydrogen bond network with the environment and within themselves also. Okay, so this is true. Since they are polar, they are hydrophilic, they can form hydrogen bonds. Last option is polar charged amino acid residues prefer to remain on the surface of the globular protein. Again, polar amino acids, they are hydrophilic, so they will be on the surface of the globular protein. Okay, so only option A. Option A is non-polar amino acid residues remain in the interior of the membrane protein. So that is wrong, because as I have explained, interior of the membrane protein is basically hydrophobic. Okay, so interior of the membrane is hydrophobic. So the membrane protein will have non-polar amino acid on the outer side and polar amino acid towards the inner side. So interior of the membrane protein will have polar amino acid, and the outer area of the membrane protein will have non-polar amino acid. Whereas in a globular protein, the outer area will have polar and the interior will have non-polar. Okay, so for globular protein, outer will have polar and interior will have non-polar. Whereas for a membrane protein, our outer will have non-polar and interior will have polar. Okay, this part is clear. So here if you see, amino acid list is there. So V is valine, L is leucine, then isoleucine, methionine, F is phenylalanine, tyrosine and tryptophan. All these are non-polar amino acids. These are basically hydrophobic amino acids. So for globular protein, they will be in the interior, hydrophobic core. This is called the core. Core means the center of the molecule. Yeah. So core means the center of the molecule. So all the non-polar, all the hydrophobic amino acids, they will be in the center of the molecule for globular proteins. But for membrane proteins, they will be at the surface. Why surface? Because the lipid is basically hydrophobic. So hydrophobic will interact with the hydrophobic parts. Okay. So it's basically like dissolve likes. Okay, so you remember that, like hydrophilic can interact with hydrophilic part, hydrophobic will interact with hydrophobic things. Okay, so if there is a lipid molecule, lipid is hydrophobic, it's non-polar, so non-polar will interact with non-polar and polar will interact with polar molecules. Okay, so polar charged amino acids like arginine, lysine, then aspartic acid, glutamic acid and histidine, they have charges on their surfaces. Okay, so they are surface catalytic sites. So if there is a catalytic site on the surface of a globular protein, they will be there. Why it is called catalytic site? So let's say I have an enzyme. In case of enzyme, the part where the substrate is binding to the enzyme, so I have some enzyme E, okay, and there is a substrate that is binding to this enzyme. So the amino acids that will be present in the catalytic site, they are generally polar, since it is exposed to the environment, it has to be polar, and they are generally charged. By charge, so that they can carry out certain reaction and this substrate could be converted into product. Okay, so if they are charged, they can transfer electrons, they can carry out some catalytic reaction. Okay, so that's why polar charged amino acids are at the surface and they are generally present in the catalytic sites. And for membrane protein, they are present in the hydrophilic core, that is the inner part of the membrane protein. Then polar neutral, so they are also polar molecules, they are also hydrophilic, but they are neutral, they don't have any charge. What are those amino acids? Serine, threonine, asparagine, glutamine, then tyrosine and tryptophan. So tyrosine and tryptophan are kind of on the borderline of being non-polar and polar, so they are considered in both cases. Sometimes they are present in the hydrophobic core, sometimes they are present on the surface. So for globular protein, they are mainly involved in the hydrogen bond networking, and for membrane protein, they are on the inside surface, because again they are polar, so they have to be on the inside surface, and mostly they are part of the channels. So there are certain membrane proteins that makes up a channel which allows certain ions and molecules to pass through the membrane. So there are sodium channels and potassium channels. So those channel proteins, they have to allow those sodium ions and potassium ions to pass through. So those residues that are lining the channel are generally polar neutral amino acids. Okay. So the next question. In most globular protein, the core is tightly packed primarily as a result of. So I have told you what a globular protein is. A globular protein is basically a soluble protein that is in a spherical form. Okay, a soluble protein that is in a spherical form. The outer part is made of polar residues. The inner core is called the hydrophobic core or and it is made of non-polar amino acids. Okay, so here they are asking about the core. Okay, so the inside the core, now I have told you the core is hydrophobic, so they have non-polar amino acids. So if you see the interactions here, it cannot form hydrogen bonds, they are hydrophobic, so this could not be the answer. There could be hydrophobic effects, there cannot be there cannot be any electrostatic effect, so this cannot be, this is ruled out. And there could be van der Waals forces. So generally in a globular protein, just a second, yeah, so generally in a globular protein, inside the core there are van der Waals forces that are present, so that is the correct answer. Van der Waals forces, basically what they do, they kind of stabilizes the whole molecule into a spherical shape. Okay, so why it is called a globular protein? It's in a spherical form, it's in a form of a globe. Okay, remember like that. So globular means a circular globular form. Okay, globe like structure. And what are van der Waals forces? These are basically weak non-covalent forces between two atoms. So if I have two atoms, uncharged atoms, if I keep them very close to each other, what will happen? There will be a slight partial charge development in both the atoms. So let's say one atom is slightly more electronegative than the other atom. So what will happen? This electronegative atom will try to attract electrons towards it, and it will develop a slight negative charge, and the other atom will develop a slight positive charge, and there will be a slight interaction between the two atoms. And that interaction, that weak non-covalent interaction, is called van der Waals forces, and they are defined by van der Waals radius and all. I will not go into the detail of the force, but then in the globular protein, mainly inside the core, in the innermost part of the globular protein, these forces are present. Okay. So the next question number 4, which of the labeled bonds show free rotation along the peptide backbone? So here a peptide bond is shown, peptide backbone is shown. We have to tell which of the labeled bonds will show free rotation along the peptide backbone. So anybody wants to answer this question?
A, 2 and 5.
2 and 5. Yes, that is the correct answer, 2 and 5. Now why 2 and 5 we will see now. So if you see a peptide bond, peptide backbone, it basically comprises of different amino acids. So if you identify the amino acids in a peptide backbone, an amino acid will have a alpha carbon, C alpha carbon, which will have the carboxylic group and the amino group. We always start writing the the peptide from the N terminal. So this will be the first amino acid, and there will be an R group. Then second amino acid will start where the amino group will make a peptide bond with the carboxylic group of the first amino acid, and then again the C alpha will be there, there will be the R group, and again the carboxylic group, and so on and so forth. So this will keep on going on, and this will make a peptide chain. Okay, different amino acids will join together through peptide bonds. I'm so sorry, just a second, something is, yeah. So different, if you see a peptide chain, you will have different amino acids that are attached together through peptide bonds. So they will form a backbone. So this is the backbone structure, N, C alpha, the carboxylic, the carbon of the carboxylic group, then again N, C alpha. And what will differ? These R groups will differ. So it could be, if it is a glycine, it would be H, if it is a alanine, it would be CH3, and like that. So the only difference would be these R group, and this is a peptide backbone that you will see. Okay. So you will see a peptide backbone here, and the R group will differ. Now there are 2-3 things that you should know. So if it is a backbone structure, if here is a backbone structure, all the bonds should be free to rotate, but it's not like that. If you see the peptide bond is basically the bond between the carboxylic group of one amino acid and the amino group of the next amino acid. The bond formed between these two groups is the peptide bond. So carboxylic group C double bond O is forming a bond with the amine group of the next amino acid. So this is the bond what I am showing. This is the peptide bond, and this peptide bond has a partial double bond character. How it is having the partial double bond character? So this amino group has a, so this nitrogen has a lone pair. So this lone pair can give this peptide bond a partial double bond character. So it can have a resonance structure. Okay. Since this has a partial double bond character, this bond becomes rigid. Okay. So now this bond, it can also rotate, but it is now kind of rigid. It has some kind of hindrance, and it cannot very freely rotate toward along its axis. Okay. Now if you see, this is a single bond, this is a single bond. Okay, this is a single bond. So all the other bonds are single bonds, so they can rotate on their axis. Now what we have here, so if you see the angle that is marked as psi, and here the angle that is marked as phi, just a second, yeah. So the angle that is marked as psi and phi, they are not the angle between the two atoms, but they are the angles between the two planes of atoms. So if you see here, this C alpha of the first amino acid is bonded to the carboxylic group, and then that carboxylic group is bonded to the amino group of the next amino acid, and this peptide bond is planar in nature. So this is one plane. The plane that is shown in yellow, that is one plane. Now this plane, so let's say I have this notebook here. So I have this notebook here. So this is one plane. Now I have another plane like this. Okay. So if there are two planes, there will be angle between two planes. Okay. So this will be the angle between two planes. Okay. So if this edge is the C alpha, then you have the carboxylic group, then this here, this will be the amino group, and then next carbon atom will be here. So it is kind of going like this. Is this structure clear? So it is kind of going like this. So the rotation that is happening is basically along the axis of one plane. So if this is one plane and this is the second plane, the rotation is happening to along the like with reference to the adjacent plane. Okay. So this part is clear. Now if you see, there is a rotation along this bond. So this C alpha, this plane, can rotate with respect to the plane that is forming with the atoms comprising of this C of carboxyl group, this C of carboxyl group, the amino N of amino group, the C alpha of the next amino acid. If you see, they are forming another plane. Okay. So this plane and this plane are basically forming a bond angle, and this plane is rotating with respect to the second plane. Okay. Is this clear? So that's how we have two planes, and then there is a rotation angle, and that rotational angle, that torsional angle, is called phi and psi. So phi is the angle between the amino group and the C alpha of the next amino acid, and the psi angle is basically the angle between the C alpha and the carboxylic group of the amino acid. So if you see in the question here, we have C alpha and the carboxylic group. So this is the psi angle, and Then we have the amino group and the C alpha. This is the phi angle. So phi is shown here also, because again this is C alpha, this is carboxylic. So here there will be a amino group. So between the N and C alpha it is phi. Between the C alpha and C it is psi. This is clear? So the correct answer is option A, 2 and 5. Bond number 2 and bond number 5 will have torsional angle. They will have free rotation to with respect to the peptide backbone. Okay, along the peptide backbone they will have free rotation. This bond omega that is shown here, it is basically the rotational angle between the peptide bond, but this is a rigid bond, so it will have lesser movement than the phi and psi angles. Okay, so this is clear. So the next question number 5. Yeah, anybody has any question? Okay, so question number 5. The angles phi, psi and omega are known as dash, and their rotational angle spans from dash to dash. Okay, so we have to fill the blanks. So x, y, z are 3 blanks given. So anybody wants to answer?
B, minus 182 plus 180.
Yes, minus 180 to 1 plus 180. The angle span is the range of the phi, psi and omega is from minus 180 to plus 180, and the angles are called dihedral angles. Why it is called dihedral angles? Anybody knows the meaning of dihedral angles?
For the angle between two planes,
Yes,
are there.
yes, very good. So angle between two planes. So just now I explained, we have one plane here and the second plane is formed by the next 3 atoms. So if you have 4 atoms, 1, 2, 3 and then 4. Okay. So one plane is formed by the first 3 atoms and the second plane is formed by the next 3 atoms. Okay. So it's basically like 2 planes attached like this. Okay, so you have 2 planes attached like this. So the angle between the 2 planes, so you can have angle between C and C alpha, you can have angle between C and N bond, but we are not talking about those bonds right now. Okay, those angles right now. So geometrically, if you have 1 2 3 4 4 atoms attached together, you can have an angle between 1 2 and 3. Okay, and you can have an angle between 2 3 and 4, but the angle between the planes of 1 2 3 and 2 3 4 is called the dihedral angle. Okay, so here the phi, psi we are talking about, as I've told you, it's the angle between those 2 planes. Okay, so that's why these are called dihedral angles, and their spanning region is from minus 180 to plus 180. So we'll look into the Ramachandran plot, the plot given by G. Ramachandran about these phi, psi angles. So there also we will see the plot is made from 180 degree, 0, and then it goes all the way to minus 180. Why it is so? Because if you see, if there are 2 planes, it can either be plus 180 or it can be the other way around, that goes to minus 180. If you have 2 planes, the maximum angle could be plus 180. Okay, you can't have an angle of more than 180 between 2 planes. So that is why the angle spans from minus 180 to plus 180. If it is rotating towards the opposite side, it will be minus 180. If it is rotating towards the positive side it will be plus 180. Okay, so basically the angle is called dihedral angle because that it is the angle between two planes, and the range is minus 180 to plus 180 because again the planes can go up to those angles. I hope this is clear. So the next question, which is the correct nomenclature for the tripeptide whose structure is shown here? So there is a structure shown here. Can you identify the amino acids and see what tripeptide it is making? Anybody wants to answer this question? So if you see here, we have amino group here. So we start counting the peptide from the N terminal only. So we have the amino group here and the chain is going on. So this will be the C alpha. Okay, this will be the C alpha. But if you see the C alpha, it is attached to a carboxylic group and the amino group, but the bond that should be formed, the peptide bond that should form between the two amino acids, it should be between the carboxylic group of the first amino acid and the amino group of the second amino acid. Whenever I am writing a amino acid, so let's say I have written a basic structure of amino acid, okay, and now another amino acid wants to form a peptide bond with this. The bond will always form with this carboxylic group and the amino group of the next amino acid, right? Right? The peptide chain always goes from N to C terminal, okay? So N terminal is fine, but then here there is no C terminal. There's the peptide bond is forming with the nitrogen itself, nitrogen and carboxylic acid. So here the structure drawn does not represent a peptide correctly. Why it is not representing a peptide correctly? What would be the correct form? The correct form should have the carboxylic group forming the peptide bond with the next amino acid and next the amino group of the next amino acid. So basically it should be amino group, the alpha carbon, the carboxylic group, and then the amino group of the next amino acid, again the alpha carbon, and then again the carboxylic group of the amino acid and the amino group of the next amino acid, and so on. Okay, so this should be the correct representation of a peptide. Is this clear?
Yes, ma'am.
Just a second. So option D is the correct answer. The structure drawn does not represent a peptide bond correctly. Next question, question number 7. The amino acid sequence of a protein is represented as LFVWSKNS. Identify the correct statement regarding the folding of the peptide fragment. So I have a amino acid sequence of a protein given, comprises of L is leucine, F is phenylalanine, V is valine, W is tryptophan, then S is serine, K is lysine, N is asparagine, and then S is again serine. So we have to identify the correct statement regarding the folding of the peptide. So let's see the statements first. First statement is fragment LFVW, that is leucine, phenylalanine, valine and tryptophan, clusters within the interior of the moiety, and while SKNS, serine, lysine, asparagine and serine again, remains in the contact with the aqueous environment. The second statement is the fragment SKNS clusters within the interior of the moiety, while LFVW remains in contact with the aqueous environment. Then the third statement is the entire protein remains as a cluster while being surrounded by water molecules. And the fourth statement is protein folding is not affected by the type of amino acid present in the peptide sequence.
I think A is the
A is so A says LFVW with clusters within the
they are
interior.
they are non-polar, so they will be outside
Yes,
the
yes,
inside the moiety.
yes, very good. So basically serine, lysine, they are hydrophilic, so they can remain in contact with the aqueous environment, while leucine, phenylalanine, valine, these are hydrophobic and they will remain inside the protein structure, so that they don't have to interact with the aqueous environment. They can safely remain uh, with without the contact of the water. Okay, so correct answer is option A. So if you see the structure of amino acid, different amino acids, you can see most if you see the side chain of these amino acids, you will see most of these amino acids can form the hydrogen bond very easily, and thus they have hydrophilic properties, whereas serine uh, whereas the non-polar amino acids like valine and then leucine, these have they have mainly aliphatic chains, aliphatic side chains. So aliphatic side chains basically make them hydrophobic, they can't make any hydrogen bonds, and thus these amino acids are present inside the core of the protein, whereas the hydrophilic uh, moieties, they can easily interact with the aqueous environment and they are at the outer portion of the protein. So this I have already shown it to you, so we can go through it again. So non-polar amino acids, they will be in the interior hydrophobic core of the globular protein, whereas polar will be on the surface or the catalytic sites. The charged will be in the catalytic sites and the neutral will be involved in the hydrogen bond networking. For membrane proteins, the things will be opposite. Now the membrane protein, the outer part is non-polar, so outer part of membrane protein will have non-polar residues, whereas the inner part of the membrane protein will have the polar residues, because they have to be protected from the non-polar environment. So they are happily sitting inside, and the non-polar is interacting with the non-polar membrane, the lipid molecules. Okay. So till now, any doubt? Anybody has any doubt? So you can ask. Okay, so we'll move on. Question number 8. In an alpha helix, hydrogen bonds are, option A is between the side chains of polar amino acids and the CO or NH of the backbone, between the C double bond O of a chain to NH of another chain that runs side by side, between the NH of I and C double bond O of I plus 4 in a single chain, and between the C double bond O of I and the NH of I plus 4 in a single chain. What will be the correct answer in a alpha?
C NH between NH of I and C of I plus 4 single chain.
C or D? It's
It starts
actually
from
it's actually option D. So if you see, a peptide always starts from N terminal. I've told you na, peptide always starts from N terminal. So if you see a alpha helix here, a peptide will start from the N terminal, the C terminal will start making, so here the peptide is starting from here, okay, and the C terminal will start making the hydrogen bond with the fifth residue of the uh, peptide chain. So let's say I have a peptide let's say I have a peptide of 8 amino acid. So the first amino acid, we will have the N terminal here, and here the C terminal will be there. So COOH, and we will have NH3 here, right? So the peptide bond is basically made between the C double bond O and NH, okay, of each amino acid. Here again C double bond O and NH. So the first hydrogen bond that will form between this C double bond O and the fifth. So if this is I, so I plus 4 means 1 plus 4, that is 5. So this will be I plus 4. Okay. So now second amino acid will make a peptide bond with the sixth amino acid. Okay. The third amino acid will make a peptide bond with the seventh amino acid, and the fourth, sorry, not the peptide bond, hydrogen bond. I am so sorry. So the first amino acid will make the hydrogen bond. The carboxylic group of the first amino acid will make the hydrogen bond with the amino group of the fifth amino acid. So here if you see, let's number the amino acids here. So here the carboxylic group is there, then there will be a C alpha, and then there will be a amino group. A peptide will always start with a amine. Okay. So I have this carboxylic group. So this is the first amino acid. Then this is the second amino acid. So this is the second C alpha carbon. So amino group, C alpha, and then carboxylic group. Then again amino group, this is the third amino acid, and then carboxylic group, then again fourth amino acid, N, C, C. So any amino acid is basically N, C alpha, and then the carboxylic group. Okay, this will be one amino acid. So this is one amino acid, then this is second amino acid, N, C alpha, C. Then again N, C alpha, C. Then N, C alpha, C. This is the fourth amino acid, fourth alpha carbon. Then again N, C alpha, C. So this is the fifth. So if you see, the first carboxylic group is making the hydrogen bond. The dashed line that is shown here is making the hydrogen bond with the amino group of the fifth amino acid. Okay. Similarly, if you see the second amino acid, this is the second amino acid. Where is the carboxylic group? Here is the carboxylic group. This is making the hydrogen bond with the sixth amino acid. Okay, so this is the sixth amino acid, and second is making the hydrogen bond with sixth amino acid. Similarly, third will make with the seventh amino acid. Fourth will make hydrogen bond with eighth amino acid. Okay. Then we have first with, yeah. So there will be, so if you see some of the amino acids that are in between, they will be involved in making the the hydrogen bond with both the carboxylic group and the nitrogen group. So if you see, for example, this sixth amino acid, you see sixth amino acid comes in between, right? So they can make the hydrogen bond. Their amino group is making the hydrogen bond with the second carbon, and their carboxylic group is also making the hydrogen bond. So all the amino acids that come in between the alpha helix, they will form the hydrogen bond with both the group, whereas the terminal amino acids, like the first amino acid is only making the hydrogen bond with the fifth amino acid. Its amino group is free. Okay, it's even the second amino acid, if you see, there is no hydrogen bond forming with this amino group. So the terminal amino acids will be free. One of the group will be free, but the middle amino acids in the alpha helix will have the hydrogen bond with both the carboxylic and the amino group. Okay, so it will always be the carboxylic group of the ith amino acid and the amino group of the I plus fourth amino acid. Okay, so this is clear. a second. Yeah. So in an alpha helix, the hydrogen bond will form between the CO of I and NH of I plus 4 in a single chain. So this kind of hydrogen bonding is called the intramolecular hydrogen bonding. In the same molecule, the hydrogen bond is forming between, like, inside the molecule. Okay. So it's called intramolecular hydrogen bonding. It is happening between the C double bond O group, carboxylic group of the first like ith amino acid with the I plus 4th residue of the amino group. Okay. This is clear?
Yes, ma'am.
So question number 9. Which of the following structure is the principal form of tyrosine? So the pK values of tyrosine is given. So pK1, pK2 and pKr is given. So R basically represents the side chain. So side chain of tyrosine is what? CH2. Then there is a benzene ring attached to a OH at the ortho position. So sorry, OH at the para position. So basically a phenol attached to a CH2 group, and then it is attached to the C alpha. So the pK value for the R group, the pK value for the amino group, and pK value for the acidic group, that is carboxylic group, is given. How do I know if it is the value for acid or base? Since the pK value is towards the acidic range, so if you know the pH scale, it is from 1 to 14. So pH scale is basically from 1 to 14. So before 7 it is acidic, after 7 it is basic. Now in an amino acid we all know carboxylic is the acid group and amino group is the basic group. So the pK1 is basically the pK value for the carboxylic group since it is acidic, and pK2 is the pK value for the amino group since it is basic, and then pKr is mentioned that it is for the R group. Okay. So we have to tell the principal structure that among these 4 structures, which would be the structure at pH 10?
D.
Structure D, you are saying at pH 10. Okay. Why?
Because at pH 10 the OH group will not be, H plus will not be released because it
Yes,
is below that
it won't be
pKr.
deprotonated. The H will not be released.
That's the 2 protons will be deprotonated.
Yes. So basically what happens, I'll explain. So we have amino group attached, carboxylic group attached, there's one hydrogen, and then the side chain. So in this case we have the side chain. Just a second. Just a second, my laptop is not functioning properly. Yeah. So for tyrosine we have the R group here. So what happens at very low pH, every group is protonated. So pH is basically concentration of hydrogen ions. So if H plus is in surplus amount, if H plus is in excess, every group will be protonated. Okay. And imagine the pK value to be a, just a second, imagine a pK value to be a line. Okay. So if the pH is towards the left side of this line, the pH has not crossed the pK, everything will be protonated. Okay. And once the pH crosses the pK value, things will get deprotonated. Okay. Remember like that. Okay. pK value is basically a straight line. If the pH is crossing the pK value, it will get deprotonated. So here we have pK given for the acidic group as 2.2. Okay. So and pH at which they have asked the question is 10. Okay. So pH has crossed the pK value. So this will be deprotonated. So it will be CO minus. Okay. Then next is the pH, the pK value for amino group is given 9 point something. Okay. So the pH has not crossed the pK value of the amino group. Sorry, pH has crossed the pK value of the amino group. So it will be also deprotonated. And if you see the pK value of the side chain is given 10. So the pH is at 10. So the side chain will be protonated only since the pH has not crossed the pK. So the pH, the side chain will be protonated in OH form, and we will have the structure D as the correct answer. Just a second. Yeah. Yeah. So here we have the carboxylic group as deprotonated because the pH has crossed the pK value. The pH has crossed the pK value of amino group also, that's why it also got deprotonated. But the pH has not crossed the pK value of the R group, and thus the R group is protonated here. So R group has not left the proton, has not released the proton yet. The carboxylic group has released the proton, the amino group has released the proton. So the correct structure at pH 10 for this tyrosine would be the structure D. In other cases, you see the amino group is here protonated, which is wrong, and the side chain is also deprotonated, which is also wrong. In this case also, so although the side chain is protonated, the amino group is also protonated. That's why the structure is also wrong, and this in this carboxylic group is protonated, so it is anyway wrong. Okay. So the option D is the correct answer. This is clear? Just a second. Okay. So moving on, question number 10. The number of atoms held into a geometric plane by a peptide bond is. So we have to tell the number of atoms that are held into a geometric plane by a peptide bond. So I have explained you a plane of a peptide bond.
Four atom. C.
4 atoms. Okay. Anybody else wants to answer? Option A. Someone has answered option A. That is 6. Okay. So let's draw a peptide bond first. So we have a alpha carbon to which carboxylic group is attached, we have the amino group attached here. Then we have another amino group of the next peptide, and then C alpha. This is a peptide backbone basically. Okay. So this is a peptide bond. So this is one. So this bond is basically forming one plane. Okay. But if you see, this plane comprises of these two alpha carbons also. Okay. So if I have a plane, just a second, I'm so sorry about this. Yeah. So if I talk about a plane, there are two more atoms that are attached with this plane. Okay. So these two C alpha, the C alpha of the first amino acid and the C alpha of the second amino acid, is attached to that plane. So that geometric plane is holding 6 atoms in total. If you count 1, 2, 3, 4, 5 and 6. Okay. Although the oxygen, carbon, nitrogen and hydrogen, these are in this plane, this carbon and this nitrogen is attached to two carbon atoms individually that are attached to this plane, and thus we have to count those atoms also, because the question is the number of atoms held into the geometric plane. So that plane is holding these two atoms also. Okay. This is clear?
Yes, ma'am.
Yeah. So and can anyone tell me why the peptide bond has this planar structure? I have told you the answer in the previous slides also. Peptide bond has a special characteristic.
Conjugation.
Yes.
Lone pair.
Yes, conjugation. So basically a peptide bond can have a partial double bond character due to uh, due to the lone pair present on the nitrogen. And this partial double bond character, this is basically a conjugated system. A double and single bond, alternating double and single bond is a conjugated system. Okay, like you have in a benzene ring. So you have a resonance structure, and that resonance structure actually makes the molecule planar. Okay, so the CONH bond lies in one plane. So if you have let's see, my hand is one plane, so the oxygen, carbon, nitrogen and hydrogen will lie in on this plane, and the C alpha, these two C alpha, they will protrude out of the plane. So one C alpha will be protruding from this side and the other C alpha will be protruding from that side. So that's how you have one plane, that is the peptide bond, and then two C alphas held by this plane on the two different sides. Okay, so that's how you have 6 atoms. So the C alpha, the oxygen, the carbon, the nitrogen, hydrogen and the C alpha of the next amino acid. This is clear? So you have 6 6 atoms attached to the plane. Just a second. Okay. Next question, question number 11. Given are the structures of two kinds of beta strands and the hydrogen bonding between the strands. Identify the correct correlation between the strands and the arrows. Arrows in the same direction indicate parallel strands, and arrows in the opposite direction indicates anti-parallel strands. So if you see, there are 4 different parallel and anti-parallel beta strands are given, one of which is the correct representation. Can anyone find the correct answer?
C.
C. Yes, so C is showing the first pair to be anti-parallel and the second pair to be parallel. Okay, so let's take up one by one. The answer is correct. The correct option is C. So first we'll see what is a parallel and anti-parallel uh, beta strand is. So if you see here, we have two peptide chains written. Okay, so one peptide chain, we are always counting the peptide chain from the amino side. Okay, that make it a rule. So if I'm seeing this peptide chain, I'll count from this nitrogen. So this will be one amino acid, N C alpha, and then carboxyl group. Then again this will be the second amino acid, then this will be the third amino acid, and so on. Okay, now let's see the bottom strand. The bottom strand has, again I'll start counting from the nitrogen only. So this will be the first amino acid, this is the next amino acid, then again N C alpha, C carboxyl, this is third next amino acid, like that. Now if you see the hydrogen bond shown with the red color, the hydrogen bond of this amino group is like the amino group is forming a hydrogen bond with the carboxylic group of the next strand, the adjacent strand. Okay, the same amino acid, so this is one amino acid, right, N C alpha and C. The carboxylic group of that same amino acid is forming the hydrogen bond with the amine group of the same amino acid in the adjacent strand. In that case it is anti-parallel. So if it is anti-parallel, so this is one amino acid, right, N C alpha C. Here also this is one amino acid, N C alpha C. So the amino group of this amino acid is making the hydrogen bond with the carboxylic group of the next amino acid, adjacent amino acid, and the same amino acid, the amino group of that same amino acid is forming the hydrogen bond with the carboxylic group of the adjacent amino acid. So both the amino and the carboxylic groups are forming the hydrogen bonds with the adjacent amino and the carboxylic groups of the, with the adjacent amino acids, carboxylic and the amino group of the same amino acid. Okay, so in that case it is anti-parallel. Now let's see in this case. So in this case we have this amino acid N C alpha C, this is one amino acid. Here also we have N C alpha C, but if you see, the amino group of this amino acid is forming the hydrogen bond with the carboxylic group of one amino acid on the adjacent strand, but the hydrogen bond that is forming between the carboxylic group and the amino group is not between the same amino acid. So this is one amino acid. Let's mark this. So this is the first amino acid, right, N C alpha C. This is the second amino acid N C alpha C. Then this is the third amino acid N C alpha C. Here also if you see N C alpha C, this is the first amino acid. Then again this is the second amino acid. Then this is the third amino acid. Now if you see, the amino group of the second amino acid is making the hydrogen bond with the carboxylic group of the first amino acid. Okay, and the carboxylic group of the same amino acid is making the hydrogen bond with the amino group of the third amino acid. So it's two different amino acid, 1 and 3. The hydrogen bond of the same amino acid, second one, is forming between the first and the third amino acid of the adjacent strand. Okay, so in that case it is parallel. And if the hydrogen bond, so here what we had the first amino acid, second amino acid, and then third amino acid. And here also if I write first, second, and then third. So both the amino carboxylic hydrogen bond and the carboxylic amino hydrogen bond, both of these hydrogen bond were between the same amino acid. The second amino acid is forming the hydrogen bond with the same amino acid. Okay, so this is anti-parallel. Okay, so this is the difference between parallel and anti-parallel. So this part is clear. So now in the options we will see. It's not going to the next slide, just a second. Yeah. So if you see in the option A, the hydrogen bond of this so this is one amino acid, okay, NC alpha C. So the hydrogen bond of this carboxylic group is forming with this amino group of the amino acid, and then the same amino group, the amino group of the same amino acid is making the hydrogen bond with the carboxylic group of the same adjacent amino acid. But here it is showing it is parallel. So this is wrong. Okay, since they are making the hydrogen bond with the same amino acids, it is anti-parallel actually. If you see here, it is the opposite. The hydrogen bonds are not with the same amino acid, and they have shown it to be parallel, anti-parallel, whereas this should have been parallel. Similarly, if you see here, the hydrogen bonding is with the same amino acids, and they have shown it to be anti-parallel, which is correct. But if you see here, the hydrogen bond is with different amino acids, and still they have shown it to be anti-parallel. This should have been parallel, right? So this is also wrong option. Then if you see the third option, we have the hydrogen bonding with the same amino acid, and it is shown to be anti-parallel, which is correct. And here the hydrogen bonding is with different amino acid, and it is shown to be parallel. This is also correct. So option C is the correct answer. If you see in option D also, the hydrogen bonding is with the same amino acids, and this is shown to be parallel instead of anti-parallel. So you get my point, right? So if the hydrogen bonding is between the same amino acids, it is anti-parallel. If the hydrogen bonding is between the different amino acid of the adjacent chain, it is parallel. Okay, so parallel beta sheets and anti-parallel beta sheets. This is clear. So option C was the correct answer for this question. So moving to the next question, question number 12. One side of a 30 residue alpha helix faces the hydrophobic interior of the protein. If the helical wheel of this helix were to be drawn, which of the following residues are likely to be hydrophobic? So let's see first what a helical wheel is. So a helical wheel is basically, you have a wheel where we place every residue of a alpha helix after 100 degrees. Okay. So, I will have a circle, and then let's say I am having the first residue here, then this is approximately 90 degrees. So, after 100 degree we will have the second residue, then again after 100 degrees, so from here somewhere here we will have the third residue, then again after 100 degrees we will have the fourth residue, and so on. So, like this on the helical wheel we place the different residues of a peptide chain after each residue is placed after 100 degrees on a circle. Okay. So, if you divide a circle, this will be 0 degrees, then this will be 90 degrees, this will be 180 degree, and then this will be 270 degree, and again 360 degree. Okay. Like so on and so forth, you can divide the circle into angles, right? So, if I am placing first residue on 0 degree scale, then this will be around 100 degrees. Okay. If this is 90, then this is around 100 degrees. Then again this will be around 200 degrees. This will be around 300 degrees. This will be around 400 degrees, and so on and so forth. It will keep on going, right? So, here in the question we have 30 residues. Okay. Now, for 30 residues we will have 30 residues placed like this. Okay. And once we have all the residues on a helical wheel, we can actually see the residues that are closer to each other. They will be part of one side of the helix. So, if I have a alpha helix like this, okay, and let's say this part is aqueous and this part is towards the inner side of the protein. Okay. So, all the residues that will be on this side of the protein, on this side of this alpha helical chain, they will be polar, right? And all the residues that will be on these parts of the chain, they will be non-polar. This is clear till now, I hope. Like something that is on the towards the aqueous environment, they will be polar. Something that is away from the aqueous environment, they will be non-polar or hydrophobic. So, this will be hydrophilic, this will be hydrophobic. Now, when you draw the residues of the alpha helix on a helical wheel, all the same kind of residues come towards the same side. So, if I draw a line here, so I can say that 1, 5 and 2 are part of the same type. So, this could be the aqueous part and this could be the hydrophobic part or the non-polar part. So, like that we can see. So, now in this question we have 30 residues. So, we will place all the 30 residues and then start seeing whether we have these residues together or not. Okay. So, if we continue placing this, so I had 1, 2, 3, 4, 5, then 6 will be 500 degrees. So, 360, then this will be, so this is 400, then 500 would be somewhere on this axis. So, 6 would be somewhere here, then 7 would be somewhere here. So, if you see like that, just a second, someone is entering. Yeah. So, if you see here, I have drawn a helical wheel for your reference. So this is the first residue. Here we have the first residue, then the second is here, then third is here, fourth is here. So like that. So if you see the first option, what is the first option we have? We have the first option as 1, 6, 11. So if you see on this wheel, first residue is here, sixth residue is here, so they can still be on in the same group, but 11 is here. So these residues are far apart, so they can't be of the same category. If this is let's say this is hydrophobic, this is hydrophobic, this cannot be hydrophobic because the range could not be that big because it's a alpha helix. So almost half, or like around half, 40% of the residues will be hydrophilic and 60% of the residues will be hydrophobic. Like that only the distribution will be there. Okay, so you can't have like 90% of the residues are hydrophilic and 10% of the residues are hydrophobic. That cannot happen in an alpha helix. I've explained you why. Because you have a helix like this, so half of the residues will lie towards the aqueous environment and half of the residues will go away from the aqueous environment, so they will be hydrophobic. Okay, so first option we rule out because 1, 6, 11 could not be in the same group. Then if you see the second option, 1, 4, 7, 10, let's see, 1 is here, then 4 is here, okay, they are still close, 7 is here, 10 is here, then the next option, sorry, 1, 4, 7, 10 and then 13. So 1, 4, 7, 10 and then 13. So these are very, again, this is a very broad range. Almost the whole circle is getting covered. So this could not be of same group. So we rule out the second option also. If you see the third option, 10, 20, 30. 10, 20, 30 will definitely be far apart. So 10 we are seeing somewhere here, 20 is here, but then 30 would reach somewhere on the opposite side of the wheel. If you keep on marking all the residues, you will find 30 would be on the opposite side of the wheel. So they cannot be of the same family. If you see the fourth option, 1, 5, 8, 12, you see 1, then 5, then 8, 12. They are very close to each other. 1, 5, 8, 12, then 15, 19, 22. Let's see. 15, where is 15? 15 is also here. 19, 19 is also here. 22, 22 will also be here. So these are of the same group. So if I now draw a line from here to here, we can say that all these amino acids are actually part of the same group, and according to question, these are hydrophobic. That means these amino acid residues are actually hydrophilic. That means if I have an alpha helix, so, and this is the aqueous environment, so these amino acids that are lying on this region, okay, all these are basically these residues, okay, and the amino acids that are lying on this region are basically these residues, okay. So that is how you can determine. So helical wheel is very important actually, because this is how you can determine whether in a helix residues will be hydrophobic or hydrophilic, whether the residues will face towards the outer part or the inner part. So when you are doing structural biology, when you are studying a protein structure, helical wheel analysis is actually very helpful in that case, okay. So the correct answer for this question is option D. I hope everybody understood how to make a helical wheel. You could be asked how to make a helical wheel. I have another question also to discuss regarding the same topic. If you have any doubt on this, you can ask me. I can again explain. So you have to just keep drawing each and every residue after 100 degrees. After 100-degree angle, you have to draw the next residue. And once you, so there might be a question where you have been given a helical wheel already and you would be asked to write the sequence of the peptide. In such cases, you will have to see, okay, like you see the first amino acid, the first, after the first amino acid, just go 100 degrees. So the 100, after the first amino acid, the 100-degree turn will give the next amino acid. So first amino acid, then after that at 100-degree position, whichever amino acid will be there, that would be second. Then again you go 100 degrees, so 200 degrees from the start, 100 from the second, that would be third amino acid, and so on and so forth. You have to name each amino acid. So if the numbering is not given, if I have, instead of writing 1 2 3 4 5, if I would have written let's say serine, then tryptophan, tyrosine, like that I have written, and I would have asked you to give me the sequence of the peptide. So you would have to start with the first amino acid and then go in the order of 100 degrees. So after one, this will be the second, then this will be the third, then this will be the fourth, and like that you have to see. So here I have drawn the helical wheel for up to 22 amino acids. You can try making it till 30 residues also and see whether all these residues are coming on the same side or not. Okay. So that could be a task for you also, and you can practice making helical wheels. Helical wheel analysis is very important in case of structural biology. Okay. So if you have to determine whether the amino acids are lying on the outer or inner side, it is very important to see. Another thing is, now I have told you in a membrane protein, the amino acid residues are non-polar, right? Hydrophobic. So if I have single alpha helix spanning the membrane, what do you think the helical wheel will show? Will it have a division like this? If I have a membrane protein, okay, this is a membrane with the lipid polar head group and lipid bilayer, and I have a alpha helix spanning the membrane. So all these residues will be of the same family, or there will be a division in the helical wheel like this?
Same family, non-polar.
Yes. So in that case, the helical wheel that will form with different amino acid residues, they will be of the same family. In that case, you cannot make a division and say half of them are hydrophobic, half of them are hydrophilic. Okay. So it depends on the type of protein that we are talking about. If I am talking about a globular protein, then yes, you can make a division like this. But if I am talking about a membrane protein, then we cannot have this kind of division. Okay, so you have to see what kind of protein we are talking about, and that's how you will have to decide. Okay. Okay, moving to the next question. Which of the following structure is the principal form of glutamic acid at its isoelectric point? So I have told you what a isoelectric point is in the last class. Again I will explain. So the pH at which the amino acid will have neutral charge, that is charge 0, that is its isoelectric point. Okay, so at that particular pH, the net charge on the amino acid will be 0. Okay, now tell me which of the following structures has the net charge 0? So if we see option A, we have no charges on the amino acid, so we can say net charge is 0. Option B, if you see, there are charges on the amino and the carboxylic group. Okay, someone has written option C. Yes. Okay, we'll see. So option B, we have the amino group and the carboxylic group charges, and the net charge is 0. Option C also we have net charge 0, and then option D we have a negative charge. So we can rule out the option D first. Now I have told you, if we have an amino acid and if the pH is crossing the pK value of the functional groups, basically the functional group will get deprotonated. Okay. Now if we have glutamic acid, the most electronegative atom will get deprotonated very easily. Okay, just a second. Yeah, so the most electronegative atom, so here carbon is attached to 2 oxygen, so this is the most electronegative, like this proton is the most, this proton is attached to the most electronegative atom. Okay, so this proton will be released first. Then next we will have to see which is more electronegative, if this is more electronegative or this is more electronegative. Obviously oxygen is more electronegative than nitrogen, so the proton of the side chain will be released next. Then at the end the proton of the amino group will be released. Okay, so this is how a titration curve of glutamic acid will go. So if we have a fully protonated state, in that case we will have NH3 plus COOH for the carboxylic group and COH for the side chain carboxylic as well. Okay, so first this proton will, so in that case we will have a +1 charge on the amino acid. So I'll draw that. So if I have a fully protonated form, that is NH3 plus COOH, and then the side chain is also protonated. Okay, so this is the scenario where at a very low pH where everything is protonated and the charge is +1. Okay, charge on this molecule is +1 from this amino group. First deprotonation will happen and this proton will leave. So basically we will get this structure. Okay, so this is basically charge 0. Okay, so from +1 we go to 0. This is charge 0. Then after this after this stage we will have a structure where this proton will also be gone. So whoever said option C, actually option C is not the correct answer, because if you see in option C, this proton is released but this proton is still attached. Do you understand? Oh sorry, option C is B. Okay, then it was correct answer, my mistake. I was seeing the structure C. So structure B is basically the correct answer, where your amino group and the carboxylic group is charged and the net charge is 0. Why? Because first the proton that will be released from the whole glutamic acid structure would be the carboxylic group's proton, which is attached to the alpha carbon. This is the most electronegative this is the proton that is attached to the most electronegative atom. So this will be released first, and then next this proton will be released, and then at the end this proton will be released. So correct structure is the second structure, B structure, that is the option C. So option C is correct. I'm sorry. So I hope everyone understands the isoelectric point concept and all. If there is any doubt you can ask me. So if I give you the pK values of all these groups and ask you to calculate the pi, that will be basically the pK average of the pK values between which we are having this neutral species. Okay. So if I have a +1 form, then I have a 0 form, then I have a -1 form, then I have a -2 form. Okay. So these 4 forms are possible for glutamic acid, right? So this is +1, then you will have this 0 form, then -1 basically where 2 carboxylic groups are deprotonated. So -2 + 1, that is -1. And then when this is also deprotonated, this is also deprotonated, everything is deprotonated, then it will have a -2 charge. So since 0 is coming between this and this, so in that case the pi would be equal to the average of the pK values for this group and this group. So pK value of this and pK value of this. So the pi will be equal to pK1 plus pKr by 2. So that's how you calculate a pi. Okay. So in this case the pi was in this case you were just asked to identify the structure, but in other cases if you are asked to calculate the pi also based on the pK values, you have to calculate like this. Okay. So at any point if the pH is lower than the pK values, the groups will be protonated. If the pH crosses the pK value, the groups will get deprotonated. Okay. And the first group that will get deprotonated would be the most electronegative group. Okay. Most acidic group, most oxidized form will get deprotonated first. Okay. And the most reduced form will get deprotonated at the end. So generally the amino groups get deprotonated at the end. The carboxylic groups are more electronegative, more acidic in nature, they get deprotonated first. Okay. They have a easier access to release the protons than the amino group. Okay. Since amino groups are basic, they cannot release the proton that easily. Any doubt? Sorry. So the next question, question number 14. From the diagram provided, find the correct statement. Okay, so we have 4 statements given and here's the diagram. So we have alpha helix and beta sheets here. So we are asked, A represents an alpha helix at the C terminus. Second statement is B represents a beta turn between antiparallel strands. C represents an adjacent parallel beta strand. And then D, fourth statement is A represents an alpha helix at the N terminus. So anybody wants to answer?
D, B, 2 and 4.
2
Option
and
B.
4. So 2 and 4 are the correct statement. Let's see. So the second statement is B represents a beta turn between anti-parallel strands. So that is very clear. We can see these 2 yellow strands, they are anti-parallel to each other. One arrow going down, one arrow going up. So these are anti-parallel to each other. So B represents the turn between those anti-parallel strands. So that is a correct statement. The first statement, A represents alpha helix at the C terminus. How do we know that is not the correct answer? How do we know whether it is N terminus or C terminus? Why is the fourth option correct? Joseph answered, right? Can you tell us why is the fourth option correct?
By looking at the arrow of the beta turns, but the arrow starts from the N terminal.
Yes. Yes, that is the correct answer. So I have told you, for any protein, whenever we are writing a peptide chain also, we always start from the N terminus. So if you see the arrow of this beta strand, so This is the start of the protein. So this is the N terminus. Then at the N terminus you have a alpha helix represented by this blue colored helical structure. Then you have the beta strands. So these two beta strands are anti-parallel to each other, and there is a turn that is attaching these two beta strands. It's labeled as the B. So second statement is correct and the fourth statement is correct. The third statement, C represents an adjacent parallel beta strand. So if you see, C is not parallel to the adjacent strand. The adjacent strand is pointed upwards, whereas C is pointed downwards. That means C is showing a anti-parallel, C is representing an adjacent anti-parallel beta strand, right? It's not parallel to the adjacent strand. So that's why the third statement is wrong. So the correct statements are second and the fourth statement, that is option B. So A represents alpha helix at the N terminus and B represents the beta turn between the anti-parallel strand. Okay, this is clear? Okay, so I have added one helical wheel question so that you have the concept clarified. So here let's try to generate the helical wheel of the peptide sequence that is given here. Okay, so M is methionine, then L is leucine, then glutamine, serine, methionine, valine, and so on. Okay, so I have drawn a circle here with the angles 0 degree, 90 degree, 180 degree and 270 degree. So if I ask you to place the amino acids on this helical wheel, can you do it? So I'll start with the first amino acid. So M will be placed here. Then the second amino acid after 100 degrees. So this is 90. So let's say this is 10. So L is here. Then again 200 degrees. So 180. So this will be 200. So Q, like that. Next will be 300 degrees, right? So 270 plus 30. So here somewhere we will have S. Can you make the complete circle like that? I would request all of you to try it out on your notebooks also. Then only you will have the practice of making these helical wheels. Then again we have the fifth residue, that is M. So it will come somewhere here. Then we have valine, V. So we will, I think, so this is 1, 2, 3, 4, 5. So we will come somewhere here. Then 6, 7th will be serine, S. Okay. Then again let's number this 1, 2, 3, 4. Wait, I have made some mistake, I think. 1, 2, 3, 4. Okay. Then 5, 6, 7, 8, 9 and 10. Okay. So this is 1, this is 2, this is 3, this is 4, this is 5, this is 6, this is 7. Now the 8th residue, where will it go? 8th residue will be somewhere here. Okay. What is the 8th residue? L. Then 9th residue will be here. So what is the 9th residue? We have L again. Okay. So this is 9th, and then the 10th residue Q, that will be somewhere here. Okay. So if you see, we have all the non-polar amino acids on one side and the polar amino acids on other side. So serine and glutamine, these are what? These are hydrophilic. So if I draw a line here, all the polar amino acids are on one side and the non-polar amino acids are on other side, right? So like I've told you earlier, if I have an alpha helix with aqueous environment on one side and hydrophobic environment on other, which part will come on the aqueous side? This part, right? This part will come on aqueous side. Why? So you see serine, glutamine, these are polar residues. So this is basically the aqueous side of the helix, and this is basically the hydrophobic side of the helix. Okay, so that's how you make a helical wheel, and you can determine which residues will go on what side of the helix. Okay, so I've taken this, like I've taken a small part of this peptide only, up to 10 residues, and I had made. So here the 18 residue peptide has been shown on the helical wheel. So after every 100 degrees you have the residues. So I was telling you, if I give you this wheel without the numbers, without these 1 2 3 4 labeling, just methionine, leucine, isoleucine written like that, and I ask you to generate a amino acid sequence like this, MLQS. If I ask you to write the amino acid sequence from 1 to 18, so in that case you will have to see, like after 100 degree which, so methionine after 100 degrees which residue is there? Leucine is there. Then again after 100 degrees what is there? Glutamine is there. Then again after 100 degrees serine is there. So like that you will have to see and then keep on writing M, L, Q, S, and then that's how you will generate the 18 residues. So if you see in this wheel, all the residues marked with yellow, most of them are non-polar, and all the residues marked in green, they are polar. So basically this is the hydrophobic part of the alpha chain, and this is the hydrophilic part of the alpha chain, alpha helix, sorry. Okay, so this is clear. Okay, coming to the last question. Left-handed alpha helix allowed region is present in which of the following quadrants of Ramachandran plot? So if you have a Ramachandran plot, which quadrant will represent the left-handed alpha helix?
Second quadrant.
Second quadrant. Anybody else? So the correct answer is first quadrant. If you see, this is the Ramachandran plot. So you have the left-handed helix on the first quadrant. So how is this Ramachandran plot made? Basically, I have told you about the phi psi angles. So we had the C alpha carboxylic group, then amino group of the next amino acid, and then C alpha of the next amino acid, like that, right? So the angle between C and C alpha plane, that was the psi angle, and the angle between the N and C alpha was the phi angle. So different angles. So let's say if you see this point, okay, this point on the graph, so somewhere around 40 degrees, so this is, and somewhere around, like let's say 10 degrees, if I have the psi angle to be 10 and phi angle to be 40 degrees, I can say that particular peptide bond will land in the left-handed alpha helix region. Okay, so like that, if I plot each of the phi psi angles for a protein and get all the points in concentrated in this region, I will say this particular peptide chain is likely to form a left-handed alpha helix. Okay, if I plot the phi psi angle plots for a peptide chain and it lands in these regions, okay, I would say that the peptide sequence is likely to form a right-handed alpha helix. Similarly, for this region, I would say beta sheet. So, first quadrant of a Ramachandran plot where both phi and psi are positive values. This is the first quadrant, right? And first quadrant will show left-handed alpha helix. The second quadrant that has the negative negative phi values and positive psi values, they comprise of the beta sheet. The third quadrant that is showing the negative phi and psi values, they are basically the right-handed alpha helix. Okay, and some part of beta sheet also comes in this, but then mostly right-handed alpha helix. So you should at least remember where which secondary structure is lying. So this is a plot that is given for determination of secondary structure. Okay, so if I have a Ramachandran plot for a protein, I can assume that okay, this protein is likely to make beta sheets, this protein is rich in beta sheet, or this protein is more alpha helix than beta sheets. Okay, like that. So you should remember which quadrant represents what. And if I have a if I have a Ramachandran plot, if I give you a Ramachandran plot, you should be able to tell me whether the peptide sequence is representing alpha helix or beta sheet or both, like that. Okay, so these kind of questions will be very relevant to this topic. So I would suggest go through the Ramachandran plot, how the Ramachandran plot was made, what is the phi and psi angle, what is the meaning of dihedral angles, the angle between two planes, then the isoelectric point, the concept of isoelectric point, the concept of pK, pH, what do you mean by pi, at what pH the charges are neutral. So those concepts you should be very thorough when you are going through these topics. Okay, anybody has any questions? So till now, whatever I have explained in the whole class, do you have any question? So even the concept of helical wheel, just try to understand. If you still have doubt, you don't want to ask right now, you can go, I will post the videos online and you can go through the video, and you can go through the slides also to understand the concept better. Okay, okay, we'll share the notes and with you. Is it still not uploaded on the portal, the problem solving session for the previous session? Okay, so NPTEL team will take care. Actually, we are just the TAs, so we don't have that information. We have shared the videos with them, so they will be uploading it soon, I guess. Okay, so if I get any information, I'll give it to you regarding the upload of the videos. Okay, Joseph has a question. Is there any difference between the red and blue region of Ramachandran plot? What do you mean by the red and blue region? So do you mean the red and yellow region? Like the two different parts that are shown here? So actually, when the Ramachandran plot was plotted, so it was basically like Ramachandran had made a model of different amino acids, and then based on the angles that were possible. So the white region that is left out, it is sterically not possible. That's why it is not involved in any of the secondary structures. Okay, so if I have two planes like this and the R groups protruding out, So there will be some steric hindrance at certain angles. Okay, so those certain angles are not included in the graph. The yellow region is basically, they are somewhat possible, they are not. The red region is showing the confidence level of the graph. So basically, if the angles are lying between the red region, they are definitely going to be beta sheet. But if the angles are lying in the yellow region, there can be some amount of steric hindrance, and like there's a chance that that angle is not possible for some particular protein or for some particular amino acid residue. Okay, so that is basically based on the confidence level of the angles. They have made this, like they have separated the regions. So in case of right handed helix also, if you see, the red region is more like it is showing more confidence. Like if you have the angles placed between those regions, they're definitely right handed helix. There's a chance that if they are in the yellow region, there is a chance that it might not be sterically possible. Like there might be some kind of steric hindrance in case of some or the other amino acid or in a protein chain. Okay, so that's how they make the graph. Any other doubt? Okay, the notes will be shared to you, I think, soon. Okay, the video of this lecture will also be uploaded. Okay, so if there are no more doubts, I will end this session now.